For 20 years, researchers hunted for the mechanism that makes trauma-focused therapy work in veterans. The answer was sitting in a completely different department, in clinical trial data for anxiety disorders. Same molecule, same receptor. The military trauma researchers never looked there. Veteran PTSD is notoriously treatment-resistant. Standard therapies fail more often than they succeed. The question that haunted the field: why do some brains stay locked in trauma while others release it? Researchers spent decades looking inside PTSD itself, scanning traumatized brains, measuring stress hormones, building models of fear circuits. The signal they needed was being recorded elsewhere, in trials for generalized anxiety, in depression studies, in obsessive-compulsive disorder research. Nobody connected them. When an AI mapped the connections between these isolated fields, something emerged. Late-stage clinical trials on anti-anxiety compounds, completely separate from military research, had already solved the implementation problem. The mechanism was serotonergic plasticity, a receptor system that temporarily unlocks rigid neural patterns. The same receptor, the same molecular key. Tested extensively in anxiety patients while veteran treatment programs struggled for answers. The bridge was there, 92% confidence. The research concepts overlapped perfectly. Psychedelic-assisted therapy, treatment-resistant PTSD. Two literatures, same solution, different filing cabinets. But then there is this. Same receptor family, 3 different molecules. Psilocybin reduces suicidal thinking. MDMA reduces suicidal thinking. LSD increases the odds. Same target system, same class of compounds. Opposite outcome on the most critical safety measure imaginable. The same receptor that unlocks healing in one molecule somehow unlocks danger in another. No one has explained why. The evidence does not merely disagree. It tears itself apart at the exact point where lives depend on the answer. The current model holds this tension without resolving it. 68% of the evidence converges on a single mechanism: serotonergic plasticity gates, neural flexibility. Temporarily dissolves the boundaries the brain builds to protect itself. This appears across psychedelic states, near-death experiences, deep meditation. But the directionality remains undetermined. Does plasticity cause recovery? Or does recovery merely involve plasticity? 3 experiments could break this model entirely. Block the receptor while allowing the experience. Measure whether the clinical change still occurs. If it does, the entire framework collapses. The answer that waited 20 years in the wrong department may itself be incomplete. This is what it means for the field to hold contradictory evidence. The solution to veteran trauma was already documented, just never translated. And the mechanism that enables healing may be the same mechanism that enables harm. The difference between breakthrough and breakdown may not be the molecule. It may be something we have not yet learned to measure. For 20 years, researchers hunted for the mechanism. The answer was sitting in a completely different department. The question now: what else is waiting there? 10 unrelated fields found the same signal, and none of them were looking for it. Psychedelics, near-death experiences, meditation, trauma therapy, different researchers, different methods, different assumptions. Yet when the data mapped onto a single receptor system, 68% of their findings collapsed into one mechanism. The serotonin system appears to gate something neuroscientists assumed required years of therapy to unlock.